Very good evening, students. So in yesterday's class, we have completed up to mitotic division. So today, good evening, students. So in yesterday's class, we have completed up to mitotic division. So what are different phases to be there in mitosis? And they will ask for 5 mark question. I've already told you so that information also. So today we'll start with this last another bit is there. So we'll start with this meiosis. Next division is called meiosis. So usually meiosis means you already know that. So this is commonly called a reductional division. Commonly called reductional division. So you already know that why we are calling it as reductional division. So why? Because of the chromosomal number reduced to half at the end of this meiotic cell division. That's what the name reductional division. So usually where you will find this mitotic division. So like that only here sites of meiosis. Sites of meiosis. So usually where you will find this meiotic cell division. So what are different sites to be there in animal cell as well as in plant cell? So when you will come across in plants. So usually in plants this meiotic cell division you will find in higher vascular plants, including we can call it as commonly called bryophytes, pteridophytes, gymnosperms, and also you will find in angiosperms. So these four togetherly called vascular plants. So in detail you have studied in botany. So in these vascular plants also you will find meiotic cell division. So for example you can take sir, you are talking about only plants. What about animals? So in animals means this is very important point. You have to concentrate more on this animals parts. So in animals, meiotic cell division you will find usually in testis and ovary. Singular I am writing. Testis means usually singular we can call. So testicles means together, or tests we can call. So testis means singular, and single in females we can call it as ovary. So in testis, what's happening there? So production of sperms going on. So you should write it down there. So production of sperms. So how these sperms are producing? So in similar way in ovary, producing egg. So usually in adults, females of reaching maturity, so they'll undergo, they'll have this regular continuous menstrual cycle. So in each menstrual cycle, releasing of one egg takes place. So in exactly 14th day of this menstrual cycle. So how this egg is releasing from ovary? So that is also by undergoing this completion of meiotic cell division. So in similar way, sperms you can take. So during this sexual intercourse time, so what will happen? So usually production of after reaching maturity in males also same after reaching maturity. So production of millions together sperms takes place, and these how million together sperms are to be produced within the testicles means by undergoing this meiotic cell division only. So that's what. So this in animals. So usually gametes formation we can call. So male gamete is called sperm and female gametes are called egg. So in similar way, so this together called gamete formation, usually in females as well as in males. So formation of gametes by undergoing this meiotic cell division. So these are all related to exactly where we are finding this meiotic cell division. So after that completion, you should know about discovery. Know about discovery. So discovery in the sense for this first time who found, so this coined the term meiotic cell division. So or otherwise who studied for the first time this meiosis. So who studied? So you write it down, Farmer and Murray. First time who studied? This is also very important point. You have to underline this. Farmer and another scientist name is called Murray. You just underline, this is very important. Farmer and Murray for the first time. So they explained about this meiotic cell division. Meiotic cell division. So this is related to sites as well as this discovery of meiosis. So directly we'll start with this meiotic cell division. So usually meiosis involves meiosis involves two processes. So two divisions. So meiosis 1 and meiosis 2. Write it down there. So meiosis will have two division process. Meiosis 2. So first we'll start with this meiosis 1. So meiotic 1 division involves two processes. So number 1, karyokinesis. Karyokinesis 1 followed by cytokinesis 1. Cytokinesis 1. And finally meiosis 2 is followed by karyokinesis 2. Karyokinesis 2, then followed by cytokinesis 2. Cytokinesis 2. Totally how many divisions you are finding in meiosis cell division? So total 2 divisions. So first one is called meiosis 1 and second one is called meiosis 2. So in first meiosis 1, that involves two main important processes. In yesterday's class only, I have already told you that to avoid that confusion, these different processes, that's what I have given that number Roman 1 and Roman 2. So usually in mitotic cell division, that also involves two main important processes. So what are those? Karyokinesis followed by cytokinesis. In similar way, in meiosis 1, karyokinesis 1, cytokinesis 1. In that finally meiosis 2, karyokinesis 2, then followed by cytokinesis 2. So this is very important information, how many divisions and what are different processes to be involved in that meiosis 1 and meiosis 2. So after that, so we'll start with that meiosis 1, then followed by meiosis 2. So first heading you note it down, meiosis 1. So this meiosis 1 involves first karyokinesis 1. You already took karyokinesis 1. So in this karyokinesis 1 involves 4 phases mainly. So first one is called prophase. To avoid this confusion, so they have written as Roman 1, prophase 1. Second one, metaphase 1. Involves 4 phases here also, metaphase 1. Third one, anaphase 1. Anaphase 1. And fourth one, telophase 1. So here also we will find interkinesis, but that interkinesis, why we will call interkinesis is nothing but that the division process between meiosis 1 and meiosis 2. Before that also one more interphase we will find. So before starting this meiosis 1, but that interphase involves only this prolonged S phase. So why prolonged S phase? So there only this duplication process takes place. If this G1 phase is there, that is very short, and then G2 phase is very short or it is absent. Actually here in interphase, before starting this meiosis 1, so interphase is little shorter than compared to this mitosis interphase. So after interphase is followed by prophase 1, metaphase 1, anaphase 1, then followed by telophase 1. So first we'll start with this prophase 1 of meiosis. So in this prophase 1 of meiosis, so diagrams to be available, you can easily understand. So this is the diagram of prophase 1 of meiosis. So totally how many phases you will find in prophase 1? Again in prophase 1, this is the diagrammatic representation. So in this prophase 1, again 5 sub-stages you will find. 5 sub-stages. 5 sub-stages where in prophase 1 of meiosis. So what are those? First one is called leptotin. Second one, zygotin. Third one, underline, these are very important. So why? Because they may ask for 2 mark question. What are different sub stages you will find in Prophase one of meiosis. So, or for 5 marks means explain the different sub-stages to be involved in prophase one of meiotic cell division. That's what leptotene, zygotene, pachytene. Fourth one, diplotene, and fifth one, diakinesis. So, what are different things to be happening? What are different events to be happening in prophase one of meiosis? Prophase one, after completion of this diakinesis only, so that will enter into the metaphase one, then followed by anaphase one, then followed by telophase one. So in this only 5 substages you are finding. In one line information I'll give you what's happening in that particular substage. So first, when you will take that leptotene stage, so here they have shown this leptotene stage. So in this leptotene, so they have shown condensed chromosomes are not that much condensed. So they are like a thin and a thread like. Along with that, in leptotene, one important event to be taking place. So which is that event? So that event is called chromosomes will look like beaded like appearance. Underline this word. So beaded like appearance. So this beaded like appearance we can commonly called a chromomere. Underline. So these beaded like appearance. Beaded in the sense, for example, you can take this is one single chromosome. This is a one sister chromatids you will find in that chromosome, and this will have dotted like structures like this dots you will find. So these dots are nothing but, so this is looks like a thread, and within that thread those dots will looks like a beads. So that rudraksha we can call. So due to that reason only, so we can also call it as chromomere. Are you getting my point? So chromomere, on what basis we are calling? So chromomere is based on, so this chromosomal threads will have that beaded like appearance. So that's for the name. So this is first event to be happening in that leptotene substage of prophase one of meiosis. Then followed by zygotene stage. I'll write here about zygotene. So zygotene, in the sense here in diagrammatic representation they have shown, so synapsis is occurring. So synapsis in the sense pairing of homologous chromosome will begin. So how this pairing of homologous chromosome? I'll just draw, you should remember. So this is the one chromosome with sister chromatids, and this is another chromosome with sister chromatids. So this is pairing we can commonly call. So pairing of homologous chromosome will begins in this phase. So this is what we can call it as a pairing of homologous chromosome, or we can also call it as a synapsis. Synapse is different. Synapse in the sense we have studied in neural control and coordination. So here we can call it as synapsis. So synapsis is nothing but pairing of homologous chromosomes. So once they will pair, so that will forms a complex. So underline this word, so many times they asked in NEET exam. What they call this complex? This complex is called a synaptonemal complex. Synaptonemal complex. We can call it as synaptonemal complex. Underline that word. So this is very important events to be occurring in that zygotene stage. So first, chromomere will get formed in the leptotene. Still chromosomes are thin and thread-like, so beaded-like appearance. Then followed by zygotene. So zygotene involves a synapsis and formation of synaptonemal complex. So these two events to be taking place in this particular phase. Then followed by third important substage, we can call it as pachytene stage. So next you write it down. This is very important. So pachytene. So in this pachytene, two different events to be occurring. So what are those? We'll discuss one by one. So in this pachytene, so here in diagrammatic representation also completely like this happened. So crossing over and recombination is taking place where in pachytene stage. This is very important. So in pachytene stage, you note it down. Two main important events to be occurring in this. So first event is called crossing over, and second event is called recombination. Second event is called recombination. So recombination in the sense, means once again they are combining. So after that, this crossing over. Crossing over in the sense what? So exchanging of the genetic material between the two homologous chromosome. In that condition, so this will forms a tetrad. This is what we can call it as tetrad condition. So tetrad in the sense, here two sister chromatids and these two are called sister chromatid. Always where exactly this crossing over is occurring? So crossing over is occurring between the two non-sister chromatids. Always you should remember, crossing over always between non-sister chromatids. Underline, this is very important. So where exactly this crossing over takes place means between two non-sister chromatids. So this is very important. So two events to be occurring. So how this between two homologous chromosome without breaking, how that the genetic material will get exchanges? Is it or not? So that's what that exchanging of the genetic material, we need that certain enzymes. So these enzymes to involved in breaking. After that exchange of genetic material, once again they will patch that whatever broken part will be there. So for that, some enzymes to be essential. What are different enzymes to be involved in that breaking as well as rejoining? So for that, enzyme name is called recombinase. This enzyme is very important. Recombinase. So this recombinase enzyme will be involved in breaking during that exchange of the genetic material. For that crossing over process, this enzyme is essential. And another enzyme is called reunion enzyme. Reunion in the sense, once that crossing over occurred means once again they have to patch their that broken part. So in that particular chromosome, homologous chromosome, so that is to be patched by or reunion by that another enzyme is called R protein. So both are very essential. So you have to remember compulsorily R protein along with that so recombinase enzyme. So these two different events to be occurring in that Pachytene stage. So after completion of this Pachytene stage, so you should concentrate more on that next stage. This is up to Pachytene. Diagram also there. You have to concentrate on this diagram also. So after recombination, so next step is called in Prophase One of Meiosis, diplotin. Here they have shown here diplotin stage. So what's happening in that diplotin? Next, the fourth stage is called diplotin, or we can also call it as a diplonema also. So both names are same. So diplotin or diplonema. So what's happening in that diplotin stage? So here chiasmata formation after crossing over and recombination. This is one chromosome homologous and this is another homologous chromosome. So after recombination, everything over in that Pachytene stage. After recombination, here this portion we can call it as chiasmata. So this will form like this X-shaped area. So this part is called X-shaped area. That's X area. We can call it as chiasmata. So that chiasmata slowly, so at the end of this diplotin stage, it will slowly both the homologous chromosomes, they will separate like this, and finally at the end of this diakinesis, both after exchanging genetic material, both homologous chromosome pair get separated. So this is related to, so what we'll call it as a diplotin stage. So chiasmata formation, and this chiasmata from the center, it will start moving towards this peripheral ends. After that, complete separation process takes place. So this is related to diplotin stage. And after that diplotin stage, and last one more substage is there, diakinesis. Here you can easily differentiate in diagram. So here near center, and but here towards that peripheral end. Towards end, this is moving towards end. So and then finally in diakinesis process, so this is exactly what happened in that prophase of mitosis. Same thing will be happening here also. What's happening? So in this chromosomes, they will start becoming thick and short. So but there only like this we have drawn, but here sister chromatids we should draw. Homologous chromosome we can call. So these homologous chromosomes at the end of this diakinesis process, so here you write it down, diakinesis. dikinesis. So in dikinesis, first thing what's happening? Terminalization. Terminalization means what? So like this chiasmata is coming towards this terminal end, peripheral end. That's what the name, terminalization of chiasmata. Terminalization of chiasmata. So this is very important. Terminalization of chiasmata takes place. So once chiasmata starts terminalizing, so chromosomes condensation begins. So here also condensation of chromosome. Condensation in the sense chromosomes, they'll become thick and short. They'll become thick and short. Along with that, one more event to be occurring in that dikinesis. What's happening? So at the end point, so you should remember, so what's happening at the end? Terminalization of chiasmata, condensation of chromosomes, thereby they'll become thick and short. So after that, finally disintegration or disappearance of nuclear envelope. Disappearance of nuclear envelope. So this is the final step. So that may lead to disintegration of nuclear envelope. Chromosomes, they already become thick and short. They will come and start lie on that equatorial plate. That is nothing but the beginning of metaphase 1 and end of the prophase 1. So in diagram also I've already shown in this. So this is what end of the dikinesis process. At the end of the dikinesis is nothing but end of prophase 1 of meiotic cell division. So after that, so directly we will move towards next metaphase 1. What's happening in that metaphase 1? So in metaphase 1, a diagram is essential, you can easily understand. So here it is. So this is the meiosis 2. Exactly this meiosis 2, before starting this meiosis 2, so this is overall prophase 1, what happened in that at the end of prophase 1 they have shown, and then followed by metaphase 1. So what's happening in that metaphase 1? So in this metaphase 1, you should note two main important points. So there what happened in mitosis metaphase, same thing here also happening, but only thing is here tetrad condition means chromosomal thread number will differ than compared to mitosis metaphase. So here what's happening? So metaphase 1, so here centromere. So centromere, they will move towards their opposite pole. They already moved, and these we can call it as a spindle fiber formation also completes. So and these completed spindle fibers will attach us to kinetochore. So in yesterday's class only, I have already told you this is what we can call it as kinetochore. So kinetochore is nothing but it is a disc-like area present in that centromere. So on both the sides you will find on the centromere this kinetochore attached to the kinetochore. So then followed by, so they will come and lie on that equator plate. So here also same thing will be happening. So in metaphase 1, they will come and lie on that equatorial plate. Equatorial plate, we can also call it as metaphase plate. So at the end of metaphase 1, same there also same, here also same. Only thing chromosomal thread. So that chromatid number may differ. So then followed by anaphase 1. So in anaphase 1, there also same, here also same. But in anaphase 1, what's happening here? So in anaphase 1, so already formed chromosomal threads are there. Here asteroids will find along with that centriole. They already move towards their opposite poles. So what's happening here? Chromosomes, so they are moving towards their opposite poles. So but here what's happening? Chromosomes exactly like this you will find. This is what I am drawing. Prophase 1, anaphase 1. So in similar way, normal anaphase when you will take, so normal anaphase it will looks like this, V shape, L shape or J shape. So this is what I have drawn. So this is related to anaphase 1. So there also same thing happened. They move toward chromosomes, move towards their opposite poles by splitting of centromere. In anaphase 1, here also same thing will be happening by splitting of the centromere. So chromosomes, they moving towards their opposite poles. So this is what I have shown. This is related to meiosis 1, anaphase 1. Anaphase 1 of meiosis 1 we can call. So then followed by telophase 1. Telophase 1 in the sense, so here two daughter cells are formed. How these two daughter cells are formed? So at the end of this telophase, so this once again reappearance of nuclear envelope. Here also same thing. Two daughter cells I am drawing now. Two daughter cells, nuclear envelope. This is plasma membrane. Plasma membrane. Here also another daughter cell, plasma membrane. Within that you are finding nuclear envelope. So then followed by chromosomes, again they will become decondensed. Decondensed in the sense they looks like a thinine thread like. Looks like a thinine thread like. This is end of the telophase 1 of meiosis. So but there only one difference is chromatid number, and along with that another very important difference you have to concentrate: metaphase mitosis telophase, then followed by meiosis telophase, meiosis 1 telophase 1. What is the difference? Here chromatid number, along with that here one more important thing you should remember. So two daughter cells will have n number of chromosomes, but there at the end of this mitotic cell division, so there at the end of telophase, there also two daughter cells are formed, but both the daughter cells will have equal number of chromosome as there in the parent cell, but not like that in this cell. So usually in meiotic cell division, so during gamete formation at the end of meiosis 1, so two daughter cells here also forming, but these two daughter cells will have n number of chromosome than compared to this parent cell. This is what the big difference between this meiosis 1 and mitotic cell division. So this completes meiosis 1. Then we'll start with this meiosis 2. So meiosis 2 exactly it is same. So it is same as that of the mitotic cell division. So mitotic cell division, what happened there? Two daughter cells are formed. So these two daughter cells, once again they will start this meiosis 2. What will happen? So this is what diagram I am showing here. Right. So this is the meiosis 2. Right. So this is the meiosis 2. So in this meiosis 2, so both cells will have n number of chromosomes. This will also have n number. This is the end of the meiosis 1. Two daughter cells are formed. So after that, what's happening? So both cells will undergo this meiosis 2. So when both cells will undergo this meiosis 2 cell division, so this will also undergo metaphase 2. So these are together called prophase 2. So here metaphase 2. What happened in that mitosis of prophase? Same thing will be happening here also. So here also disintegration of nuclear envelope. Here also disintegration of nuclear envelope, and chromosomes condensation will begin there in mitosis prophase. In meiosis 2 prophase 2 also same thing will be happening. So condensation of chromosome. So condensed chromosome, they looks like a thick and short. In both the daughter cells, same thing will be happening. And later on in metaphase 1, so these two will come and lie on the equator plate. In mitosis metaphase also same thing happened. Firstly, in yesterday's class I have already told you about metaphase of mitosis. In similar way, here also they will come and lie and finally form these spindle fibers are there na? So these spindle fibers will attach us to the kinetochore of the centromere. They will attaching to the kinetochore of the centromere. So after that, followed by, so both they will enter into the anaphase 1. Sorry, anaphase 2 here. This is anaphase 2. So in anaphase 2, what's happening? So the splitting of the centromere in both the daughter cells I am talking. So in both the daughter cell, this will also split like this, and it will come chromosome towards their opposite pole. In another daughter cell like this, and so they will move towards their opposite pole. So by splitting of centromere. By splitting of centromere. So this is related to anaphase 2. So then followed by telophase 2. Telophase 2, exactly this is opposite of prophase 2. What happened? It disintegrated nuclear envelope and nucleolus once again reappearing. So here they have shown small round now within that. So this is what related to. This is nuclear envelope. This is nuclear envelope. This is nuclear envelope. This is nuclear envelope. Are you getting my point? Total, how many daughter cells are formed at the end of meiosis 2? So total 4 daughter cells are formed. So these 4 daughter cells will have n number of chromosomes. All 4 will have n number of chromosomes. Why? Because of meiosis 2. I have already told you that this meiosis 2, we can also call it as, this is also equational division. Equational. So all these 4, prophase 2, metaphase 2, anaphase 2, telophase 2. So these 4 stages comes under karyokinesis 2. I've already told you once the same thing here also. In meiosis 1 also same. So in meiosis 1 also, prophase 1, metaphase 1, anaphase 1 and telophase 1. So these 4 phases comes under karyokinesis 1. Karyokinesis 1 followed by cytokinesis. Cytokinesis means same. So at the end of this meiosis 1, so here I have shown right. So here, so at the end of this meiosis 1, so totally 2 daughter cells are formed, and these 2 daughter cells, how these get separated in the sense that is by undergoing. So this is when we'll consider telophase, meiosis 1 telophase. So furrow formation takes place. There also cytokinesis 1 will be there. So furrow formation, after that furrow, so complete 2 daughter cells may get formed. So 2 daughter cells in the sense completely here at the end of meiosis 1. So n number of chromosomes. So 2n will become n, and both the daughter cells may get separated by formation of cytokinesis 1. So that is furrow formation between the 2 daughter cells. So same thing happening in that meiosis 2 also. At the end of this telophase, here also telophase, and this is another daughter cell telophase. So both telophase, this also leads to one daughter cell chromatin network. This is another daughter cell chromatin network, and this is another daughter cell, and this is another daughter cell. At the end of meiosis, so how many daughter cells we got now? So 4 daughter cells. How we got that 4 daughter cells? By undergoing cytokinesis 2. Karyokinesis 2 is followed by cytokinesis 2. Karyokinesis 2 involves what? Understood. So prophase 2, metaphase 2, anaphase 2, telophase 2. At the end of telophase 2, this daughter cell, the telophase is different, and this daughter cell telophase is different. So both they will undergo cytokinesis 2 by formation of furrow or constriction between the 2 daughter cells. After that, so these 2 daughter cells will completely separate after the formation. So total, how many daughter cells are formed at the end of meiosis 2? Total 4. So both all 4 will have n number of chromosomes. Same exactly if they ask for 5 marks question, explain about meiosis 2 division, means same. You have to take 2 daughter cell, you have to divide separately mitotically. So then at the end of this meiosis 2, so how many daughter cells you are finding? 4 daughter cells you are finding, and all 4 daughter cells will have the same number of chromosomes. So this is at the end of karyokinesis 2, then followed by cytokinesis 2. So this is related to meiosis 1 and meiosis 2. Then followed by one more important thing you should remember. What is the significance of meiotic cell division? So next very important thing you should remember. Next, significance. You've already studied in previous class what is the significance of mitotic cell division. In similar way here, significance of meiosis. Significance of meiosis. So what is the main important significance of this meiotic cell division? So first, number 1 significance we can call it as, so this will also maintain the chromosomal number, and along with that by recombination, I've already told you in Prophase One of Meiosis, recombination takes place. Recombination and crossing over. You will ask question then, sir, Why? By exchanging genetic material, what is the use in that sexually reproducing organism? So why? What is that use means? You have to remember, by recombination. Re means once again combination, new varieties will come. So that is by undergoing crossing over. So crossing over leads to variations. Crossing over leads to variations. Variations in the sense, in all the sexually reproducing organism, so parents usually differ from their offsprings. Usually offsprings differ from their parents. Why? Because of due to variation. Variation due to recombination and crossing over. That's what. So we will not exactly have similar appearance, morphological appearance to our exact copy of this parent cell. Why we will not have this exact copy of our parent cell? Why? Because of this crossing over is occurring. Due to that crossing over only, so exchanging of genetic material occurring between paternal and maternal, that's what that new developing young one will have so many variations. So by that variation, what is that use significance? So second significance, we can call it as variations may leads to variations may leads to evolution. Variation may leads to evolution. So evolution means how we come from our ancestors. So this is by undergoing a process, naturally occurring processes called evolution. So evolution is mainly, so this variation. So variation is majorly due to crossing over and recombination. So these processes will be occurring only in sexually reproducing organisms. So this is related to what is the main important significance of this meiotic cell division. So by this completes your chapter cell cycle and cell division. So by this also, so this complete all zoology chapters, right? Thank you.